Hey everyone, this is Dr. Mikola Rashik coming back to you with another video dedicated to mRNA vaccines. Today I have a great and hot topic, and the reason why is because I'm going to present to you the first evidence I have managed to come across that corroborates the idea that the spike protein might be entering nucleus. And what's even better, it's from a Pfizer documents themselves, and it's also vaccinal spike protein. So this is the second evidence we've ever seen, and it is from Pfizer and vaccinal protein. I can't wait to show it to you. We're going to jump right into the midst of this. This report was brought to my attention by a medical doctor from Australia when we were discussing the idea that so far there is no published evidence that corroborates the Swedish study that I discussed in video number 18, which was the first time we've seen this suggestion made that the spike protein could be entering nuclei and potentially, according to those authors, impair the ability, our natural ability to fix the broken DNA. Here we're going to present that second data from a document released by the Australian authorities, government authorities that are responsible for approving medications in Australia. So let's just jump right into it. So this is the actual link you can see. This is also the title of the document. It's published by the Therapeutic Goods Administration of the Australian government. That's equivalent to the FDA. That's the component of the government that's responsible for assessing the presented data on medical devices and therapeutics. And that's the organization that approves their public use. This is, as you can see from the title of this report, non-clinical evaluation report, meaning this is the data that was done on cells in vitro, meaning outside of body, as well as on animals. So this is all of the data that was presented to the authorities prior to the authorities providing agreement to Pfizer to start using these vaccinal constructs on humans. So let's jump right into the main component that I want to show you. It's on page 34. And we're going to go through this entire report in the meantime. Don't worry, it's a fantastic piece of work. So page 34, what we're looking at is section 2.3, expression of antigens. Antigen in this case refers to the spike protein itself that is being produced from the genetic material introduced via the vaccine. We're not going to describe this in detail, except that I can tell you that HEC293 T cells, those are very standard cells to use for studies in science. They are human embryonic kidney cells. They are used as a gold standard. The reason why is because this cell line is somewhat immortal, basically. We've been propagating the same cell line since 70s, and therefore it's been used extensively in science, and that's why it's used as a gold standard. That's one of the reasons why this cell line was picked for these studies. Right away, let's jump into the data. So this is the first data in this section. You can see on the left-hand side, a graph that shows how effectively the different vaccine constructs were infecting the cells, meaning entering the cells. This is called transfection in science. So basically, we're talking about the vaccine construct being able to enter the cells. First, you can see when they use the genetic material that is now used on humans in the Pfizer vaccines, when they use just the RNA itself, you can see that's in black bar, how effectively that alone was able to transfect cells, as compared to in gray bar, the actual entire vaccine product. So now we're talking about the RNA, the genetic material that's encapsulated within a lipid molecule. So lipids are fat molecules. Lipids is what makes up the cell membranes. So the film that covers the cell, that makes the cell wall in our body, that's a very thin layer of fat. Vaccine products, they're inserted into own independent little balls of fat, and this allows vaccines to be able to fuse directly with the cell membrane as they encounter the cells. And you can see from this graph that such construct of the genetic material inside the vaccine lipid construct, it transfects or gets inside the cells even easier. So that's what this data represents. And that the genetic material is the same, as I mentioned, that is used on humans. Now, it's the next data that is really the data that we want to look at, and that reveals that the spike protein might be entering nucleus. The reason why the authors did this study that I'm about to show you is to prove that the spike protein, the vaccinal spike protein, once those cells are transfected with the vaccine, meaning the vaccine goes inside, that the spike protein is produced in the cellular compartments we expect them to be produced in. In this particular instance, we talk about endoplasmic reticulum, ER, and the plasmic reticulum is where the proteins are produced, and they want to show, hey, our spike protein from the mRNA we're introducing in the cells will be produced in the endoplasmic reticulum. They did not look for anything else. So I think they might have accidentally overseen the fact that their own data shows potentially presence of spike protein inside the nucleus. So let's take a look. That's this information right here. Let's zoom in on it and let's explain what we are looking at. So let's look at the top row. The top row represents different images of the same cells that have been transfected or injected with vaccinal product. So this is, you can see the name of the vaccines. That's the same vaccine that is being used on humans, same genetic material. And at the bottom row, that's where you have cells that have not been transfected. So no vaccine was used, and you do not expect spike protein to be produced inside those cells. Now we're going to go from left to right, column by column. The first column in blue, what you see is a stain that actually stains nuclei inside the cells. So nucleus is where the genetic material is being housed. It's think of it like the brain of the cell. And this clearly shows you the outline of the nucleus within those cells. Next, in the second column, you see antibodies that have been tagged with a fluorescent tag, which allows those antibodies to fluoresce, give off signal, in this case, red signal, when you hit it with a specific wavelength. And clearly you can see this antibody is supposed to be attaching to proteins found inside endoplasmic reticulum. So this allows the authors to basically demonstrate where is that endoplasmic reticulum present inside the cells in relation to the nucleus. Next, they use the same technique, but with different color for spike protein. So they use different fluorescent tag for spike protein. So basically they use antibodies with a specific fluorescent tag. Wherever the spike protein was present in those cells, those antibodies would bind it and fluoresce and give off green signal. And that's what we see right here in the top row. It kind of shows you that spike protein is present almost everywhere inside the cells. And the bottom, you see no color, no image, because no spike protein was being produced. So you just see nothing but dark black image because now fluorescence is being detected. Now, the last column is the most important because that's where all of these signals are being merged together. So how does this work? You see in blue, that's again where the nuclei is present within the cells. Now, what you see in red, once again, represents endoplasmic reticulum. What you see in green, once again, represents where the spike protein is. And now we see additional new color, that's yellow. Where you see yellow, that's the overlapping signal between the endoplasmic reticulum, the red signal, and the spike protein signal, the green signal. And whenever you see red and green signals overlap together, they combine together to create the yellow signal, yellow color. So you can almost see that the vast majority of the endoplasmic reticulum is flashing yellow, meaning spike protein is being produced actively inside there. That's what these authors, these scientists wanted to prove. But what no one looked at, no one commented on, is the fact that you can also clearly see green signal of the spike protein when it appears right inside the nucleus, and only inside the nucleus, clearly within boundaries of the nucleus. So this is the second time I see any evidence showing that the spike protein might be found inside nuclei. What's even more interesting, we're talking about vaccinal spike protein, and we're talking about also same cell line that these authors used, that was used in the video 18 study I discussed. So authors of that Swedish study, they use exactly same cell line. So additional proof that this might be a real consideration we should be paying attention to. All right, I'm going to go over the rest of this report because this report was amazing. It was very well done. as a scientist, I was very impressed. I would have been very proud to provide such a report for anyone's analysis. So what I'm saying, both the scientists who did the report did an excellent job, and the people analyzing the report, the Australian authorities, also did a very good job. So let's talk about this. The next data that we see immediately after is really interesting as well, because it shows you some of the decision-making processes here done as to why the final vaccine product was selected as it was. So what we see here, let's focus on a left-hand side graph. We see different vaccine constructs being used to target the cells, transfect the cells, get inside the cells. V08 refers to one of the types of vaccines that Pfizer developed based on spike protein. In the end, that was not used on humans. V09 is the one that ended up being used on humans. And there's few different options that they studied. U refers to unmodified RNA, meaning the RNA chemical components were exactly the same that are normally found inside RNA, naturally inside cells. Then you see MOD, that refers to modified, because the vaccines that are being introduced to us, they use modified chemical nucleotide bases that are typically not found in nature. So they're synthetic, they are not typically used to produce RNA. As you can see, one of the reasons why this modified option is being used is because in one way, it does help to increase the transfection of the cells. It means such a construct is more easily used by the cells. So that might have been one of the decision-making processes. There's another one that they use, which is labeled as SA, that refers to self-amplifying RNA. It's a very unusual construct. In the end, was not used on humans. That's where the vaccine was coding for a little molecular robot that once produced would allow to remake more of the vaccine. So basically it would produce more of the same RNA genetic material. And you can see in a middle graph that if you transfect the cells with the same construct and you look which one of these produce most spike protein after the vaccine has entered the cell, the self-amplifying one produces most spike protein. That makes sense because obviously once you inject the genetic material, more of genetic material is first produced, and then you use that genetic material to produce more spike protein. Nevertheless, that was not what was used in the end for vaccinating humans. It was the one with the modified synthetic chemical nucleotide bases to create RNA vaccines. And that might in itself have some biological consequences yet to be uncovered that we don't even potentially know of yet. They are not the same as what is normally found in nature. Next, we're going to go and look at this beautiful image. The reason why this image is so remarkable is because, as far as I know, in the literature that I've studied, this is the first time I've actually been able to see an image of a vaccinal spike protein. So how does this work? What you're actually looking at here in panel B is think of it as an actual photograph of the vaccinal spike protein as the authors would have seen it on the surface of the cells that they transfected with the vaccine. So those cells produced the spike protein from the vaccine, and then they presented that spike protein on a cell surface, and that's what you see in panel B. How did that photograph was taken? They basically bombarded it with electrons, and wherever electrons were bouncing off, this allowed to create this beautiful three-dimensional image that you see right here. That's one of the many techniques that we can actually use in order to create extremely sophisticated three-dimensional images of proteins, including spike protein. Most of these that I've seen are for viral spike proteins, so the ones that the virus would produce. As I mentioned, it's the first time I see one for vaccinal spike protein, and they divide them into multiple different families based on slight differences in their shape. The one that is of interest to us in this particular context, if we look at the one to the furthest on the right, You can see there's a little knob sticking out. That little knob is the receptor binding domain. That's the domain or portion of the spike protein that needs to flip up. And as it flips up, it is available to interact with the human receptor, the ACE2 receptor. So we can actually see it standing up here. If you were to take a more closer look at this image, you could potentially see that it's actually more distorted than the others. It's not as well filled. And the reason why is because the movement of the spike protein in order to present that receptor binding domain actually distorts the image. So it's similar to like you trying to take an image of a galloping horse and you took a photo and you no longer see the legs because they were moving too fast. It's the same idea right here. The movement was too fast, so it distorted the image. We cannot capture the same quality image as in the other families presented by the other colors, but together all of these can be combined and we can create a very detailed image presented in panel A, where you can basically determine where every single atom is placed in three-dimensional space of this vaccinal spike protein. As I mentioned, fantastic work. This was even available to us prior to vaccines even became available for use in humans. So that's amazing. I would have loved to have seen this sooner. We're going to move forward from this. And we're going to now look at some of the very famous distribution data of where the vaccines might be distributed throughout the body. This data has been discussed numerous times previously in many different videos, because this is the exact same data that is now famous as the data presented to the Japanese authorities. And that's where the first information came out suggesting that the vaccine constructs could be entering ovaries, because you might have heard that this data presented to Japan was done on mice. That's actually not accurate. It wasn't done on mice. It was done on rats. This is the same data, except now it's available in English, so I can study it better. And the numbers are exactly the same. So whatever that Japanese data is, we're going to go over it right now in more detail. So what this data showed, that injection site was always the main site of where the vaccine was being found or concentrated. And by the way, how did they do it? They use radio-labeled, one of the lipids was radio-labeled, so they were able to see where the radioactivity was concentrating in these animals post-injection. The second most common impacted organ was liver. So you can see that data right here. That also makes sense because after all, eventually liver is one of the organs involved in breakdown of whatever is found in our body. So that's not a surprise that eventually somebody would end up in being broken down in the liver, including the lipids of the actual vaccine vehicle that is introducing the genetic material inside the cells. The other two major organs are spleen. So that's the data right here, as well as adrenal glands. And that also would make sense because those are the tissues responsible for producing some of the immune response products. And therefore, the information might be presented in those tissues in order for the immune response to take appropriate place. The one that was surprised in fifth place was the ovaries. So you can see that information. This is where that entire background information came from, that potentially vaccines might be accumulating in ovaries. We don't know if this happens in humans because I've never seen such study yet done with humans as to where do these vaccines get distributed throughout the body post-vaccination. This might make sense though. Ovaries might be targeted in humans and we should investigate that. And the reason why is because, as I discussed in video number 28, there is now emerging evidence that menstrual cycles are being affected post-vaccinations, and that's not fully understood how that happens, but this might be one of the contributing reasons, and we still yet to be testing that in humans. The only other data that I'll present at the end here of this report, before I start going back to the earlier pages, is what happened to the babies of rats that have been immunized with the vaccine. So that's presented right here under the section called fetal variations and malformations. This is the actual data. And this was somewhat interesting conclusion by the governing authority in Australia, in that, yes, there was a differences seen, so that the pups of rat mothers that gave birth to those pups post-vaccination, the pups were being more frequently affected by fetal birth defects than control group. However, and the most common problem was seeing extra ribs develop. So you can see the data right here in this table. However, the authorities agreed with the Pfizer scientists that this can be dismissed on the account that the frequency of the abnormalities observed post-vaccination was still completely within the norm of what was observed in other historical studies in control groups. So while you can see here the three different vaccines, number 1, 2, and 3, this one, number 2, is the one what we ended up using with humans. You can see that there's always some extra abnormality seen in comparison to the control groups, meaning those who were not vaccinated with anything, especially with an extra limbs. But as I already mentioned, it was concluded that that's okay, because these extra abnormalities are not outside the range that can supposedly be seen naturally in this type of rats. So that's why this was not investigated any further. And I'll show you that information as well. Before I go to the very beginning of the document, there's one more data that I'll show you, which is the antibody levels that have been observed in monkeys post-vaccination. And the reason why is because this data showed that monkeys that were vaccinated, they clearly produce an effect, and they can see there's an post-vaccination in blue, there's a dramatic rise in antibody levels. And that rise is even higher post-second vaccination, shown here at the day of 28. But then soon after, what was clearly observed was a waning effect. And they studied 2 different doses on these monkeys. By the way, all of the studies that I'm showing you on animals, the rats that I just showed you and the monkeys, the doses used are much higher in comparison to what is in the end used on humans. So in humans, this is the amount, the dose used on humans, but keep in mind, humans in general are much, much bigger per weight than monkeys. So these animals, both the rats, at least 100-fold higher concentrations were used in rats for the data I was just showing you a moment ago. Same thing, multiple higher doses were used on these monkeys. Nevertheless, despite these higher doses, clearly there's a waning effect where these antibodies are disappearing over time. And this is also shown in this graph right here, where what they measured here is the ability of the antibodies to prevent the virus from infecting cells. And you can see post first vaccination dose, there was an increase in the ability of the plasma antibodies to stop the virus from infecting cells. This increase was enhanced post second vaccination, but then over time it started to wane as well. So the ability to stop the virus was decreasing from infecting the cells, was decreasing as the level of antibodies was decreasing as well. And I'll show you right away how the Pfizer scientists addressed that to the Australian authorities. So now I'm going to go to the beginning of the document, where there's a page of all the summary statements by the Australian authorities in relation to the data that was presented to them that we just saw here. And in terms of the information I just presented to you about the antibody levels in monkeys is summarized right here. The authorities clearly mentioned we're observing waning effect. That's a potential concern of the ability of these vaccines to raise long-term immunity. And of course, now we know what history shown, but I'll show you how the scientists from Pfizer tried to address this and why the authorities moved on with vaccination, despite this observation being available right away from the start. Clearly, we've been able to observe that later on with humans, where post-vaccination, the antibodies wane, they do wane rapidly. And this is why we continuously need boosters in order to pump them back up and to reintroduce the protection against different variants. So that's this summary right here. Right above that is another important summary, and that's the fact that these vaccines that are being used, both on humans as well as used in these studies, they use novel lipids, meaning novel fat molecules that are synthetic, that are not found in nature, and that have never been studied on humans. So that's another concern that the Australian authorities raised. Once again, I'll show you right away how this went about and how scientists were able to bystep this concern. The only other comment I'll highlight on this page is the fact that the distribution of the actual genetic material, the mRNA, is not known. It's at this stage, it was not known. I don't even know if it's still known as to what happens with the distribution of these vaccines post-injection. The good news is that they appear to be disappearing fast. And as I showed you, there was the distribution on rats. They showed you where the vaccines are distributed, but it's not the same as actually tracking the vaccine genetic material. They were tracking lipids. So that's still a concern that they raised in terms of us understanding where does this vaccine actually go post-injection. We're going to move along. I'm briefly going to stop on this page. This is only to mention what these two novel lipid synthetic compounds are that we've never used on humans before. They are called novel excipients in this literature, so you might see that right away. Page 8, this is once again summarizes the concern about the waning antibodies as seen in monkeys that have been injected with vaccine. And this is how the Pfizer scientists addressed it. So this is in this paragraph right here, highlighted right now. This is how this Pfizer scientists addressed it right there. The sponsor, meaning the Pfizer company, stated that the long-term immunity was being assessed in an ongoing phase 1 studies in the US and Germany up to 2 years following vaccination. So basically, and that will provide a more accurate measure of long-term immunity. So basically, they decided to defer understanding of this till 2 years later. Well, we already know what happened, right? We already know how rapidly these antibodies do wane and potentially why we've had to resort to use of so many boosters in such a short duration time. So that was basically this. They decided to, let's find out what the data will show us later on. And they moved on. Moving on, let's take a look at novel excipients right here. This is on page 10 of the report. This shows you a little bit of information as to what happens with these novel compounds in the animals, how they get excreted from the body. One of them seems to be retained in the body for a long time. We don't know how long, because the length of it in the studies when it was looked at, it was retained throughout the entire study duration in some of the animals. However, the doses, this is where the dosing we're talking about, the dosing can be much, much higher than is studied in these animals than what is being used on humans. So that's an important distinction to make. And we're going to move on to page number 12, because page number 12 and 13 collectively, they give us the explanation as to why the Australian authorities assume that it's okay to accept the use of these novel compounds, synthetic lipids that have never been used on humans in future vaccinations, which is what happened. And the reason why is because the Pfizer scientists cleverly mentioned, hey, the structure of these novel compounds is very similar to structure of compounds already approved in other medical uses. This is the product in which such lipids, other synthetic lipids were approved and they were deemed to be safe. So because due to the health crisis emergency, we can now assume that these new chemical compounds we're introducing that are similar to the ones that have been previously approved and found safe, let's just assume that these will be safe as well. And the Australian authorities agreed, although somewhat begrudgingly, because they do mention, well, they are similar, but they are, after all, structurally and functionally different as well. Right there. So that's that one sentence right there that summarizes that. Meaning, well, it probably would be good thing to find out what do these new compounds do long-term. So that summarizes the report. That I will end right there. The take-home message right here, the big one, is that the Pfizer data on its own is showing the possibility that the vaccinal spike protein could be entering nuclei of our human cells. We should be studying this further. That's the big take-home message here. What I also wanted to let you know, folks, we're having another video survey going on. So you can check out the link to that survey in the description below. Basically, it allows you to select what video content we should be making in the future. So what interests you the most? Let us know. And that's the videos we'll be making later on. So we've done this a few times before. Let us decide once again. Also, we have another COVID Q&A event coming up. This one is now number 6. So we're still moving along, still people coming along. These are great fun. We have a number of questions from the audience that we collect from the videos or emails. I answer those questions, and then we have an open mic to interact, and anything goes. So any questions that audience might have, or if they want to present scientific information, we can discuss that as well. Lots of fun. Check it out. Link to the event is also in the description below, and I hope to see you there. That's all for now, and we'll see you in the next future video. Bye, everyone.